Salt is a primary, life-sustaining nutrient that the human body cannot synthesize internally. While modern nutritional advice frequently warns against overconsumption, maintaining strict physiological boundaries of dietary sodium chloride is essential to sustain vital life mechanisms. Far from being a simple seasoning, salt works at a microscopic level to operate cell transport systems, facilitate neural electricity, and sustain organ function, advanced fluid dynamics and osmotic balance, extracellular osmolarity management. Sodium serves as the primary solute in extracellular fluid. It exerts the necessary osmotic pressure to draw and hold water outside cellular walls, active water redistribution, operating alongside intracellular potassium. Sodium controls the cellular fluid equilibrium through constant water movement, vascular volume preservation. Dietary sodium directly preserves blood plasma volume. This ensures adequate tissue perfusion and prevents sudden hypovolemic dehydration, bioelectric signaling, and nervous system function. The sodium-potassium pump. Animal cells use up to 40% of their daily ATP energy to fuel the Na+, K+ ATPase pump. This pump actively forces 3 sodium ions out of the cell for every 2 potassium ions pulled in resting membrane potential. This asymmetric ionic shift creates a stable negative electrical gradient across plasma membranes, preparing the cell for activation, action potential. Initiation. When neurons or muscle fibers fire, gated voltage channels open to let sodium flood back inside. This generates the electrical spike that powers heartbeat control and voluntary movement, gastrointestinal secretion and nutrient transportation, hydrochloric acid secretion. Parietal cells in the stomach utilize the chloride ions from salt. They swap them for bicarbonate via an anion exchanger to synthesize gastric hydrochloric acid, HCl, protein, enzymatic activation. The resulting highly acidic gastric environment is crucial for converting inactive pepsinogen into active pepsin to break down complex dietary proteins. SGLT1 active transport. The small intestine utilizes the sodium-glucose cotransporter 1, SGLT1. This requires 2 sodium ions to bind simultaneously with a glucose molecule to force energy into the bloodstream against an uphill gradient. Physical performance and critical homeostasis. Myofibril relaxation cycle. Sodium allows the rapid depolarization needed to clear calcium away from muscle fibers. This stops involuntary muscle spasms and severe heat, cramping, thermal regulatory stability. Maintaining sodium levels helps the cardiovascular system support sweating while keeping up adequate blood pressure during high temperature exertion. Dilutional hyponatremia prevention. Adequate dietary salt intake keeps blood sodium levels from dropping dangerously during heavy endurance workouts, protecting the brain from swelling. In conclusion, ultimately, salt acts as the structural foundation for cellular hydration, electrical communication, and gastrointestinal transport mechanisms. Striking a precise dietary balance ensures that these highly integrated biochemical systems function flawlessly without overloading your heart or blood vessels.